Well, how are you?
Showered.
Oh, yeah. That's good. All right. Hello, Lalith.
Hello.
So, yeah. Away with, did you do anything? Do you have anything to follow up on from last time? Or with Susan?
Hello.
How are you?
Pretty good.
Okay.
I managed to get the first draft of my paper written about my thesis, so
Oh, good. Congratulations. What was it to? Was it to a journal?
I haven't decided.
Okay. It's just in the loaded up and ready to go. Huh?
Well, I'll get it to a journal. I don't know which ones Dr. Zhang would like it to go to, but
Okay.
I wondered if it would be good for the usual journal, Biosystems. Maybe.
Yeah.
I don't really care. Just I promised to do this properly, and I get to publish it. So, yeah. Anyways, and I would like to do some more work on it. Actually, it's just that I need a less expensive program or a more readily available one. So maybe I get to learn ANSYS.
Yeah.
I don't know. And unless they give it to me for less than $3,000 US, I'm like, no. That's kind of expensive, don't you think?
Yeah.
Yeah.
That's good. Good luck with that whole process.
Mm.
Lalith, you were going to say something.
So, we discussed about the hypergraph structures and message passing, and we discussed about there are like few things that we discussed in the last week that we have to see what could be a good data set to understand the interactions and the mechanisms and the biological interactions that they do, the causal mechanisms that each and every cell divides during the morphogenesis, and there are various biological interaction patterns. You wanted to look into some of the data sets, and in previous meeting we discussed that there were some of the works where they also included phase angle as one of the component to they incorporated into the structural features. So, and I was going through the papers, like the big list of papers that Dick has attached like last week, and I was trying to read this the work which is I can share PDF, so it's more of physiomorphogenesis. That's one of the paper he suggested. So I was looking into that, how this can be, the centrifugal cells, how we can group them using a hypergraphs and all. So I kind of have a thing that I can show you.
Okay.
So I tried off using that, incorporating the phase angle into the, our temporal lineage graph tree, and to find out the, how is that phase angle, changing when we divide the, cell from the parent to like two child. So this is what I have seen for the first division. It has around like 198.9 degrees, and later the next division it has 195, which is the next subsequent divisions, and so on. So I was able to capture this exact phase angle. So it's like a simple formulation that we use for like how do we calculate phase angle. So we can calculate the phase angle using this function. This is for the XY plane. So given so the theta is the phase angle. So this is like tan inverse of 2 times the y coordinate change and the x coordinate change. And so this we get in 0 to 360 degrees. So this is the map that we get. So x1 and y1 are like the coordinates of 2 daughter centroids. So based on that centroids, we are able to plot this. So this was for 3 time points shown. So this we can incorporate into like all the time points. So yeah, so this is one thing which was kind of little took time. And also I was looking into one of the paper that for the data sets. So what they have done in this paper is they have a lot of data set. So they have C. elegans volumetric microscopy data set consisting of like standardized format. They have pre-processed, and they have a human in loop machine learning based analysis tools also. So they have a diverse collection of 118 whole brain neural activity imaging data sets they have acquired from the 5 labs, and they store it in wormid.org. So this is their their community where they have like data sets open sourced. And how do we extract them and analyze, like what are the things that they have provided is so they have this automated cell identification algorithms, and they have recovered all the cases, and they have a repository to identify factors that influence the developmental positioning of neurons. So they wanted to identify this. So this is their task. So for this, they have like 118 whole brain neural activity images of that. So this has like an app properly developed, and the pipeline looks like this. I haven't gone to the paper entirely, but I have looked into this. This was like one of the important work that I've seen. So this is how they have. So they have designated into color spaces, and they have plotted the angle. And at each of the activity, they have this traces. They have 2 different types. This like calcium based on acquisition, and there's a neuropal acquisition. So this is like raw neural activity, and this is neuropal structural volume. So this was like what they have provided. I wanted to look into this more probably if if we are trying to understand the biological insights and all. So probably later in time, so you can look into more details about it. This data sets is available in this like for open source. So we need to spend little time into this to understand more. So I can share this paper in the group so that it will be useful to people. So this is 2 things. And I've been reading about physiomorphogenesis and how that work could be useful for our developmental embryogenesis and all. How can we form these interstitial cells together and wire them, and like the fusion, the cell membrane divides, and like certain components are like this cell membrane is like a big thing. So when it does this fusion and it divides into like 2 different things, what are the key changes that actually happen? So we can also model that.
Yeah, that would be good to have. Thank you, Dick, for all the papers that you sent. You sent probably about 20 papers.
Yeah, and I was able to find only one thing that could, I can understand a bit more. They were all more of biology and all.
Yeah. So that paper you just showed looks really interesting.
Yeah.
It's like
yeah.
Is this a new one? Like just recently?
This is a very new one that it was recently published in like cell reports methods, like 2020 January 2025. Yeah. Okay. Yeah. I think that's the 2 things. One, I calculated the phase angle, understanding the that it can also give us some more structural insights that can be embedded into all the time points. So that's one thing. And second is I was able to understand the physiomorphogenesis and read about a little more into the data sets that recently have, can be directly used for like this hypergraphs. So that the biology that we discussed, the interactions, be more. So that's it from us.
Okay. That's good. Thank you. So yeah, thank you, Lalith. So it looks like Jayadratha and Priyanka are here.
Yeah.
Would you like to introduce yourself, Priyanka?
Sure. Yeah, sure. My background, because I'm at home. So hi, I'm Priyanka. I'm doing my PhD at Kansas State University right now. I'm in my third year of my PhD. So I got to know about this project from GSOC, and I found it a little bit interesting. To give you a little bit background, I did my master's from IIT Gandhinagar in India, and I worked as a data scientist for 3 years. And then in fall 2022, I started my PhD. So right now I'm working on influence maximization as well as graph neural networks, where the idea is to build scalable and transferable system, where we not only looking at how do we represent the system, but also looking at how do we identify that how these like units in this particular network or what we are interested in interact with each other. And then I'm looking for homogeneous as well as heterogeneous graph representation as well, and some of the hypergraph work, where I'm looking at some of the causal analysis, where we are looking at why certain things affect other part of the networks, looking at the features representation of those data, as well as what cause certain things more important than other. So right now, the mostly data I work on is social network as well as infrastructures model, where I am representing different infrastructures model as part of graphs. So that is like kind of my groundwork, but mostly I'm looking at influential node in the network. So some of this work, like looking at the hypergraphs or causal analysis, excite me to work on this project. Right now, I got some idea by looking at the GitHub repo, and I played around some of the codes, but I did not had anything to directly represent now. But definitely, I'll have a look. I just want to get into touch with what's going on this meeting and what problems specifically you guys are looking at, and where it is required for us to, give some contribution.
That sounds good. Thank you. Yeah, we're kind of working on we're working on the GSOC project and sort of the ideas kind of coming out of that. So that's what we've been talking the last few weeks. So that's it kind of in line with what we were talking about last week with causal analysis. So
Yeah.
yeah.
Yeah, right now I am looking at the causal analysis, looking at the features of some of the nodes in the graph. Like for example, I have explored some of the notebooks as well, where these worms have certain kind of features which help them like maybe directly related to their growth, or how do they interconnect with each other. So that I found a little bit interesting, and maybe in hypergraph representation as well, like how do we say that something is coming to a common cluster or not. So maybe we can look at some of those features as well, which is more important than other.
Thank you.
Yeah.
All right. So this is we'll come back to this paper that Lalith posted. So this looks like a really interesting paper. I think it's from January, so it's pretty new. Looks like there are a number of people on here who only one I'm familiar with is Ev Yamini. But yeah, this is on whole brain imaging. So it's there's a whole community of people in the C. elegans world who do like connectome stuff, looking at neurons, and then there are other people who are doing the rest of the organism along with the brain. So it's kind of you're going to get, I think with this paper, you're going to get a lot of neuron information about the neurons, and of course the neurons are roughly about maybe 1/3 of the worm. So it's 302 neurons, 959 cells in the adult hermaphrodite, so it's like about a third. And we know the connectome, it's been estimated, and we know all the connections. So we have a pretty good handle on the C. elegans connectome. That's one of the reasons why C. elegans is such a great model organism, is because a lot of these features of the individual cells are known, and we have community data sets for them. So, the same holds true for the connectome. We know, like, every neuron, we know their position. We actually know the position of all of the adult cells because it's very regular from worm to worm. And so this paper goes into characterizing these neuronal cells, the determinants of their position. So they're usually positioned relative to one another. Some are anterior, some are posterior. They're neurons in the tail, they're neurons in the head, and they're neurons down the body, length of the body. So you have this nervous system that emerges in development. And so the question is how do the neurons know where to go, or how do they end up where they end up? Because it's important for, say, like tail neurons to end up in the tail, head neurons to end up in the head, and then, neurons to be near one another, because when neurons connect, that process of connection is where axons are sent out using an axon cone to find another cell or find another axon to connect with. So this is a process that, you can't have cells just at random locations in the worm. They have to be, within a reasonable distance, or with, in a place where those cells can be signaled in some way. So the highlights of this paper, so this is, I think, in Cell Reports Methods. The highlights of the paper, they produced this NWB file. This is a specific informatics file format that they've modified to extend supporting C. elegans whole brain imaging data. So NWB is something they use in neuroimaging. It's a data format that involves a number of different attributes of the neuron, the electrophysiological data, and so forth. Production of a diverse data corpus using harmonization tools at wormid.org. So this is their wormid site. Let's see if we can go through to that. Okay, it's not. I don't think it's a hyperlink, but anyways, they have a data repository there and some tools. Neuron ID algorithms improve in performance and do not need lab-specific retraining. So this is, of course, where they're developing an algorithm for doing this prediction. And then Neuronal Atlas reveals biological determinants of neuron positioning. So they are able to sort of get a handle on why a neuron is in the place that it is. And that's an interesting question, C. elegans. And actually also for this notion of syncytium. Is like, why do cells end up in the place that they do? I mean, we know that they end up in a certain place, and it's obvious that some cells need to be near one another. Certainly, if you have a syncytium, all the sort of the muscle cells, or the epithelial cells, or the germ cells that are involved in some syncytium or another need to be next to one another. I guess it's most efficient if they're next to one another. Like we discussed, you can have syncytium that are, all over, scattered, and then you can have cell bodies that sort of meander throughout the worm. But, if you think about muscle, you want to build a muscle body. You have all of these cells that divide, and they differentiate into these muscle precursors. They have their own nuclei, and then the cells fuse into a structure that resembles some sort of large, solid body that can contract and expand, like a rope or something like that. So you have this form, higher order form that you want to achieve. And in the nervous system, of course, neurons form circuits that produce behaviors. So you have to have those connections in place. Maybe you have sensory relays that need to be that can't be too distant from one another. C. elegans, the distances aren't very great, but there are these neural conduction issues if the cells are just everywhere in the body. So this is useful information. So they go through the data here. Their conceptual abstract is here. I don't know if you can open it up. Okay, let's see. Okay, here we go. So basically they have whole brain imaging of C. elegans from 5 labs using a methodology. They merge these into a standardized format, NWB, and then they work on software for harmonization and data curation. So they have NeuroPal, which is a long-standing software, and then EatsWorm, I don't know what that is. And then they retrain. So a lot of these groups will do things where they're exploring ways to build algorithms to train, train these predictive models that can give you, if you're in a lab, information about neuronal ID or behavior classification or something like that. So, you can imagine if you have some high throughput system where you're imaging a bunch of worms, you want to identify the cells, their location, and then a lot of different attributes about those cells. Say you're doing a cell tracking experiment where you're getting microscopy data from a bunch of worms. You're doing this high throughput acquisition of the images. You're getting the images and you're just kind of trying to ID stuff from maybe like a microscopy image or a series of microscopy images. And so that's what their aim is here. So they retrain 3 neuron ID algorithms, CPD, STAT Atlas, and CRFID. So these are just different algorithms for this sort of task, and they want to see which one is best. And then this is wormid.org, which is this data corpus and neuron analysis tool. Okay, so that's the sort of the summary, and then they kind of talk about these whole brain imaging experiments with single neuron resolution, which they call whole brain imaging. This is something that they've developed in C. elegans because it's small and we know all the neurons. We have a nomenclature for them. We know all the connections. So the connectome is known in C. elegans, but in a lot of other organisms they're not. So we have, for example, Drosophila, which is the fruit fly. We know some of its connectome. People have produced high resolution maps of different parts of the Drosophila brain, such as the mushroom bodies or other areas where they have a characterization of the cells and the connectome, but it's incomplete. And there is a lot more prediction there because there's a lot more of a lot less deterministic process. Plus, there's just a lot more stuff in that brain. And then, of course, we also have the demonstration whole brain imaging in the zebrafish, and that happened in 2013. And of course, we've adopted these methods for Drosophila, for zebrafish, and for C. elegans, and worked on them since. And so, it's going to be harder to do in mammals because mammals, first of all, they have this sort of regulative development where the cells differentiate according to chemical signals and gene expression signals rather than a deterministic signal. So you can have a bunch of precursor cells that could be one cell specific cell or another. The cell type is more restricted, but the ID of a certain cell in a certain location is less deterministic. And so that's something that that's why it's harder to work out lineage trees in those organisms, but also use this method. So, but they're trying, and they've tried in mice to do this apparently. Yeah, so this is really, they make this point here. Manual cell identification from fluorescent microscopy images is a notoriously difficult skill, requiring substantial expertise and labor. This task is particularly difficult for neurons labeled with nuclear localization of fluorophores, which are these fluorescent signals that you see in the microscopy images. If you've looked at some of the microscopy data, you'll notice this image that's black with a black background with all these lit areas, these like little dots. Those are the we localize fluorescent signals to the nucleus, sometimes to the membrane, but mostly to the nucleus. And you pick those up in the in the microscopy. You segment those, and you can segment the position of different cells. And you can also do this with membranes, but the localization of these fluorophores, the ability for you to segment them out from the background, that's all very tough work. It's not the easiest thing to do, and so you need to have these kind of algorithms that can help you not just segment things but identify things. So knowing the position and knowing, like, if a cell divides and its daughter cells are going to be in a certain position, that's an important piece of information, because then you can predict where a cell should end up, right? Like if you have AB, like like we were showing before, and it divides into two cells. Those cells migrate from the position of AB, and they migrate to new positions. They could be on opposite sides of the worm. They could be in different positions along the AP axis. We just have these positions that are estimated from cell tracking data, and we know from the lineage tree that there's a relative relationship in their AP position, their anterior posterior position. So we can have those kinds of estimates. So that's what they're trying to work out here. Okay, and so they introduced this tool, Worm ID. This resource consists of data harmonization tools, so like creating these standardized file formats. NWB stands for Neuro Data Without Borders, and there are actually some GSOC projects through INCF working on Neuro Data Without Borders. I'm not sure what the projects are this year, but they're always working on this format and working on how to sort of expand that format and make it useful to people across neuroimaging and in neuroscience. So that's what they're trying to do here. They do a lot of pre-processing work. They have this open source software to analyze whole brain activity images. And they also have tutorials and documentation for using these kind of tools in people's pipelines. So if you're a wet lab doing a high throughput analysis of microscopy data, you have these tools right at your fingertips, and you don't have to worry about training them and working on the computational aspects of data wrangling. This is one of the things that OpenWorm has been trying to stress over its time, is not just the simulation part, but sort of the data analysis, the data normalization part. So the idea is that you want to have people in wet labs, which is where you have some sort of worm culture facility, and you're interested in basic questions in the biology, and you don't really want to become an informatics lab. So there are a lot of informatics tools you need to do your work, and so there needs to be a set of tools, and in the C. elegans community there are a lot of great tools, but you need to have a set of tools that are reliable, that can allow you to just plug and play and find these things and not spend a lot of time on that, so you can go to those basic questions. So that's what they're trying to do here. So finally, we provide a large online corpus of harmonized C. elegans whole brain activity imaging and structural data that can be used for large-scale experimental analysis, neurobiological modeling, and algorithmic development. This corpus is stored in a popular community archive called the Distributed Archive, for neurophysiology data. Of course, this is Dandy, and I don't know if he's not here. Morgan actually is a big fan of Dandy, and he knows a lot about it. So if you're interested, you get in touch with Morgan. He can tell you about these open access repositories. Yeah, let's see. This is their, let's see if this is the one where they show some of this. So this is where we have the angle, and so we model the worm as this this volume, and it's basically an XY surface that's wrapped. So this is something that we've tried to do a lot in the group, is establish like a four-dimensional position. So what you have here is you have an XY axis. And what happens in microscopy when you acquire images from a C. elegans embryo, from a worm, I guess, too, is you have the spatiality where you have, the worm or the embryo on a cover, on a slide with a cover slip, and it's squished down a little bit. And you have this XY position, so you have left, right, anterior, posterior, which is forward and backward. And then you have the Z position. And the Z position is usually depth. So it's, where you go from the top to the bottom. And so that position, oftentimes in a lot of the cell tracking data, is estimated from different images in an image stack. So you'll image a stack through the focal plane. You'll go from like the top down to the bottom, and it'll just focus in on different layers. Sort of, you take a sphere, you take this this shape, and you cut it across, and you get these different slices of the image. And so, because it's squished with the cover slip, it's a little bit distorted, and there isn't a lot of information on the z-axis. At least in my experience, I've never seen a lot of information revealed on the z-axis, not as much as on the xy axes. So what they're doing here is they're taking the xy plane and they're wrapping it around. They have this toroidal surface, and they're actually trying to establish the angle. So you have the angle, you could have like an XY angle. So you could see like if something divides at the lower right, and it, the cells migrate upwards and leftwards, then you can estimate that angle. And you can use a two-dimensional estimate of that, no problem, like we saw with the theta value that Lalith showed. But then you have the three-dimensional aspect. And there's a way to estimate three-dimensional phase angle as well, which is where you have this, you can have like the three-dimensional position where you add in the z-axis and you figure it out. Or you have this sort of toroidal space where it's like this continuous space, continuous XY space around the edge of this space. And then you have this estimate of position, or the estimate of angle. So you have this butterfly plot, which just shows the theta value from negative 180 to 180. So that's 180 degrees and negative 180 degrees. And then showing the distances there. And you see that there's a lot of phase angles that are centered around 0, which should be expected because that's the center, or close to where the cell. I don't know if they're taking it from the origin point of the cell, of each cell, or if they're taking it just as an absolute position. But you should expect either way that cells won't necessarily diverge as much. Some cells won't necessarily diverge as much from their mother cell in terms of position, and other cells will. But it's also the case that you don't have that much divergence in position. Maybe it's like cells begin at the center line of the worm and move to the edges. Anyways, this is an interesting plot. It's interesting data, And then, of course, we have activity traces for the different neurons with a time course there. We have images of raw neural activity. So this is where they're imaging. I guess it's some sort of electrophysiological, like a voltage dye or something, where they're looking at these different things going on in the neurons. This is the neuropal structural volume, which I think is a good measure of volume for the cells. They just stain, I guess, the whole cell for this. So this is available in the adult. Maybe they have this for development now, I'm not sure. But this is where they have the structural volume, I guess, for the adult cells. So this would be where we have some estimate of that. We also have these, we do have, in this case, the z-axis and then the y-axis, so the dorsal-ventral view, which is the z-axis, and the lateral view. And then we have these color space views, which we can get into. So, what do we talk about? So yeah, so there's a lot of interesting data in here. Why don't we talk about this data source for worm ID? I'm interested in that. So this is worm ID. This is where, this is very user friendly, I guess. This is where we're IDing your worm neurons. This is where we have just the neurons, of course. So we don't have every cell in the adult. We have the adult cells, which is they're the terminally differentiated cells in the lineage tree. So at the bottom of the lineage tree, when you have a bunch of developmental cells that divide and then they end up at a state where they don't divide anymore, they don't differentiate anymore. And this is what they have in this. So ID or worm neurons, worm ID org is a resource for the C. elegans neuroscience research community to do all the things that we talked about. We have data sets here, which is on this tab. So we have, let's see, we have their Dandy ID, the data set initials, and then the description, and then they have a source lab. So these are, I think, 7, right? Yeah. And so the first one is from the Hobart lab. This is neuropal microfluidic chip images and GCAMP activity. That's the one where we saw at the bottom right of that last figure. This is where we have like 21 worms that they sampled. You have a number of segmented neurons. So they're not complete, connectomes, but we get like a pretty good representation of neurons in each data set. So this is 166 to 188 neurons. And then this is, their notes over here where they kind of take data from other sources. So you can go back to those sources and see if you can. So there's just a lot of data that we can use for this. Some of these are unpublished, some of these are published. So, but the point is that they're available and we can get access to them. Yeah, so this is good stuff. There's a lot of whole brain neural dynamics, whole brain imaging, these neuropal structural volumes. So this last one is from the Cato lab at UCSF. 10 worms, 166 to 180 neurons. And, this is unpublished data sets from the Cato lab. So this is, I don't know actually if these are available, but in any case, those are the data sets. This is the code. So this is the Neurodata Without Borders repository for C. elegans. This is, where they have the Neurodata Without Borders formatting files. So this is just for people to convert their optophysiology data. So this is where they're doing like things like gel or voltage dyes and things like that, and they're able to convert that to this format. Okay. Yeah, so then they have apps. So they have EatsWorm as a Napari plugin. So Napari is a imaging tool. And then NeuroPal, of course, is something you can use on your it's a standalone application you can download. Yamini has been developing this for a long time. So, and the reason that's important is because he's been involved sort of peripherally with OpenWorm. So this is good. And then you have how to use, and you can go through and basically set up neurodata without borders and then extract the activity using et.al. So that's what that paper's about. I think that was a very good paper. And Morgan is here now, so he's usually in transit at this hour, so he probably wouldn't be able to speak to it. But it's just nice data sets available on Dandy to whatever extent they're available, and they're worth checking out.
So that's a good presentation. So thank you for the detailed understanding of the paper. So I was trying to understand the components more thoroughly. I thought of reading that and bringing out next week, but it was fortunate enough to get it this week. Yeah. So yeah, that's from my side.
All right. All right, so I think we'll finish up here with, let's see, which paper do we want to do here? We'll do this paper here. This is, I can't remember if this is something that Susan sent. This is kind of shifting to a different topic here. But this is the flow analysis of the low-Reynolds number swimmer C. elegans. So this is Physical Review Fluids, is the journal. And this is the abstract. So, and when we talk about flow analysis, of course, we're talking about when something is floating in water, it's moving through water. We want to get a sense of its Reynolds number, which is the sort of the dimensionless parameter of its ease of motion through that flow. So you could have a laminar flow, you could have a turbulent flow, and then different things that move through those flows have a Reynolds number, and the Reynolds number tells you something about the hydrodynamics. So that's what we mean by like a low Reynolds number swimmer. It's moving through this water column. The water column has like a certain level of turbulence. It could be laminar, which means it doesn't have any sort of turbulence, meaning that it's, there's no sort of, there are no cross currents or anything. And we're talking about very small amounts of water. So it's, not like we're talking about huge volumes of water. So, but that's, we want to get this Reynolds number because we want to see, what the shape of the body is relative to the environment that the organism is in, or if it's a cell, what kinds of, what does the cell need to move through that water in terms of a motility mechanism or something like that. So this is an interesting question for like biophysics, for mechanics and things like that. Okay, so the abstract reads, swimming cells and microorganisms are a critical component of many biological processes. In order to better interpret experimental studies of low Reynolds number swimming, which is where the Reynolds numbers are really low, the flows are laminar, and you have this, the swimming where they have to move through the water column, we combine experimental and numerical methods to perform an analysis of the flow field around the swimming nematode C. elegans. So C. elegans is usually in some sort of liquid. It's usually some suspension. It could be what, in nature, usually C. elegans are in these environments where they're in the soil, but they're also in a very wet soil. So there's liquid, there's colloidal material around them, and they have to find their way through. So that's why this is relevant. And then if they're in culture, they're usually on a gel, or they're usually swimming through like maybe a bacterial soup. So that's another reason why this is relevant. So, and they do have these swimming modes where they are in water and they kind of wiggle back and forth, and they have these various swimming behaviors. So this is also why this is important, because we want to know kind of what kinds of flow fields C. elegans experiences in movement. So this has relevance to the muscles. This has relevance to the output behaviors and how to keep their behaviors stable so that they can swim and get places. We first used image processing and particle tracking velocimetry, which we've talked about that method before. We haven't really talked about it in depth, but there's this method where it's like cell tracking, where we track the nucleus of the cells, except in this case, we're treating things as different particles, and we're tracking their velocity. So we're not just taking the position, we're taking the derivative of the position. So this is in a way kind of like these phase angles, because you're taking particles and you're calculating out their velocity, their movement, or changing position over time. And a couple weeks ago, I did a paper, this was after the main meeting, on these Lagrangian coherent structures that people have used in development. So Lagrangian coherent structures are where you have, you use particles to track movement. So you'll have a bunch of particles at an origin point, and those particles will disperse. And you have this technique, you construct this Lagrangian to track the cells from their origin point and how they diverge from one another. And then you can calculate out these ridges where you have, you can make statements about their movement together, and you can make statements about the flow. You can make statements about the agency of the things that are moving, et cetera, et cetera. So there are a lot of Lagrangian methods we can bring to bear to this type of thing. So we first use, we did the particle tracking. So they use particle tracking velocimetry to extract the body shape, kinematics, and flow fields around the nematode. So again, we got the body shape, we have the kinematics, the movement, and then the movement, and then the flow fields around the nematode. We then construct a three-dimensional model using the experimental swimming kinematics and employ a boundary element method to simulate flow fields. So we need to also simulate the flow field so we can get the Reynolds number, obtaining very good quantitative agreement with experiment. So they construct a model, and then they compare model with experiment. So they measure things, and then they build the model, and then measure the model, and they see how close they can get. We use this numerical model to show that the calculation of flow shear rates using purely planar data results in significant underestimates of the true three-dimensional value. So this is where, again, we have this issue between the two-dimension and the three-dimensional model. So we can represent things in an XY plane. And, a lot of our microscopy is acquired in XY planes. And then we do, the modeling in XY planes. And that's, one way to do that. But then you also have to sort of extrapolate that out to 3 dimensions. And so when you use planar data, which is like the 2-dimensional plane, it's easier computationally, but you suffer from not being able to make a true estimate, especially in 3D, because anything when it moves through a water column will move in 3D usually. You'll have all sorts of things that will move in a water column. They might move up the water column or across, and they'll have variations in terms of, in XYZ space and how they move. So there are significant underestimates of the true 3-dimensional value. And then this leads us to the 4th-dimensional model, because the 4-dimensional model is time. So we want to understand this stuff over time. So the variations in time, there's acceleration and deceleration in time, and so forth. Applying symmetry arguments validated against numerics, we demonstrate that the out-of-plane contribution can be accounted for by incompressibility, and therefore simply calculated from particle tracking velocimetry. Our results show how fundamental fluid mechanics considerations may be used to improve the accuracy of measurements and biofluid dynamics. Okay, so this is good stuff. So they'll talk a little bit about why this is relevant to C. elegans. So they talk about swimmers. There's, of course, mammalian spermatozoa, which are, very small cells that have flagella, and they travel using these undulations. So they fluctuate back and forth, and they move through the water column. So there are a whole class of cells and organisms that are these undulatory swimmers. One such undulatory swimmer is a biological model, C. elegans. The genetics and physiology of this nematode are well studied. Its genome has been completely sequenced, and a complete cell lineage has been established. The neuromuscular system of C. elegans controls its body undulations, which allow it to swim, dig, and crawl. And then C. elegans has also been used as a model system for experimental studies of propulsion, particularly at low-Reynolds number, due to its simple planar swimming gait and size. So this has actually been used in propulsion studies, which are, 9 through 15. These citations down here, they don't have the name of the papers, but a lot of soft matter and fluid physics and stuff like that. Okay, so that's what they're doing there. So we have this sort of model that we can build. The nematode generates planar bending waves through contractions of its ventral and dorsal muscles. This is why we're interested in the planar representation. So when you have this wiggling back and forth, it generates this planar set of waves, as you could imagine, and it contracts ventral and dorsal muscles, and it produces this quasi two-dimensional traveling sine wave along its body. So anterior to posterior is this sine wave at around 1 millimeter in length and 75 microns in diameter. So it's very small. C. elegans is significantly larger than the majority of low-Reynolds number undulatory swimmers. So a lot of the undulatory swimmers are cell sizes of several microns. You're talking about like spermatozoa. You're talking about E. coli, other bacterial cells. So we have this large multicellular system. So it's much larger than the other types of undulatory swimmers. And so we can study these sine waves. They're bigger, they have larger dimensions, and Reynolds numbers and flows behave differently at different size scales. So the closer you get to the nanoscale, the more different it is. And so these smaller undulatory swimmers are kind of closer to the nano regime than C. elegans. So what we know about these systems is, kind of limited down to that size scale. So we have a little bit different size scale to deal with. The resulting flow fields can be used to probe properties of both the swimmer and the fluid, providing new insights into the physics of undulatory propulsion. So, and then of course, just because we have this planar representation we can build and this swimming stroke that's planar, the flow around C. elegans as a complex three-dimensional structure, which is difficult to capture experimentally. Then he used flow properties such as the shear rate and are dependent on out-of-plane flow contributions not measured by planar velocimetry data. In essence, one measures a two-dimensional slice of a three-dimensional field. So we have that limitation. This limits, for example, our ability to accurately determine drag and propulsion forces experienced and exerted by the swimming nematode. In this work, we show that the numerical and theoretical fluid mechanics techniques can be used to improve the processing and analyses of these experimental flow fields. So let's see if we can find some images. So this is Figure 1. This is the set of instantaneous flow streamlines generated by C. elegans. So they've computed this from experimental waveform data, where they've calculated this in the media. So this is where you see this complex three-dimensional flow field. You see things happening along the, laterally. You see things happening in this sort of C space, and you see the strength of the flow in different places. So the closer to the body, the stronger it is. So you can see that this is not a simple sort of wake that it produces. It produces a lot more. And then, of course, if the fluid is turbulent, then it complicates things even more. Because I think this is assuming a quasi-laminar flow. I don't know what this is just kind of like, I think this is a very simple case. This is where they have the skeletonized body, which is here at the left. You have the reason they do that is because they just want to capture the kinematics. They're not interested in the cells. All they want is the shape of the body and then this kinematic skeleton, so you can see where the degrees of freedom of movement are, and you can model those appropriately. So you see there are many degrees of freedom along this axis. And some, when you have an undulation, some of the worm is moving right, some of it's moving left, and it keeps switching position as it undulates. So it's going back and forth. So you have these experimental settings where you observe this, you observe the kinematics and the direction of the movement, and then you have this skeleton that you can build and simulate it. So you simulate it in a flow field, which is here, and you see this. So this is particle tracking velocimetry. And this shows the flow field produced using the reconstructed 3D mesh and a numerical model. So this just shows the speed in terms of millimeters per second. So you see at the head where there's a change in direction, it's faster. At the middle of the middle of the midline, right here, it's also faster. And so actually the places where it's switching immediately where it's switching direction, you see it slower. So these are just basic mechanical characterizations. Okay. So this is the spatial distribution of the experimentally measured x component, which is uxy, simulated x component, which is in B. So we have the experimental measurement in A, the simulation in B. And C, we have the experimentally measured y component, Vxy, and then the simulated y component, which is Vxy simulated. So you have experimental simulation for x and y components. And so this is where, again, we have our color bars, which are these component velocity magnitudes, which are millimeters per second. Again, we see differences. So the x component, you see it towards the head and the tail. For the y component, you have it around where it's actually changing position or changing direction, and also at the sort of the end of the head and the end of the tail. So there are different components here for this. So the spatial distribution is different for the x component and the y component. Okay. Then this shows the comparison of flow structure between experiment and simulation. This is A is the distribution of U, B is the distribution of V, which goes back up to the x and the y components. Then there's distribution of speed, which is absolute value of U, and the decay rate. So you have the distribution here of U, V, the distribution speed. So this is speed instead of these spatial components. And then you have this decay rate. Comparing experiment and simulation. So, we try to get the simulation as close as possible to the experiment. And we have some loss in distribution speed at the high end of the speed, and then the middle here of this decay rate. And, some differences in terms of the spatial distribution as well. So this is kind of what they were talking about, where the experiment and the model are not exactly aligned. And then this is the shear rate error calculation and correction. So this just shows like this error, and they're dealing with it here. Okay, so then the conclusion is that they use theoretical and numerical techniques to improve the processing of experimentally obtained particle tracking data, producing smooth velocity fields, quantifying the flow around the swimming nematode C. elegans in a Newtonian solution. So this is Newtonian fluid, Newtonian paradigm. So you could use, I guess you could use non-Newtonian fluids and get a different result. It's much more complicated to compute, etc. We compared our results with a 3D boundary element model of the nematode, generated directly from experimentally obtained nematode kinematics, finding good agreement between numerics and experiment. We argued that when calculating derivative flow quantities, the only non-trivial out-of-plane component was WZ, the z derivative of the z flow. So this is where we have this, I guess this is I don't know what an out-of-plane component is, but I guess this is the thing that was throwing off the model. So it's the z derivative of the z flow. So it's just basically, higher order kinematics. This observation was validated by simulation, showing that excluding WZ when calculating the flow shear rate results in a significant underestimate. Accounting for WZ component via incompressibility from 2D data eliminated this error. Our work illustrated how theory can be used to improve experimental measurement in biological fluid mechanics, will be directly applicable to the investigations of biolocomotion in complex fluids, like in nature, where they find themselves in fluid flows, maybe with colloidal things around them, maybe in a complex three-dimensional plane that they have to travel, etc., etc. Furthermore, we anticipate that as technology for the acquisition and processing of experimental flow fields continues to improve, these out-of-plane effects will represent a hard barrier to increasing the accuracy of results, making such techniques increasingly important. So this is where we're getting more data on experimental flow fields, and then we have this out-of-plane effect that people are going to run into when they try to do a plug-and-play type model, and they're going to have to figure out why that is. So as we gain data, as we improve our ability to measure those things, we have to be able to model them and then maybe hand them off to someone who doesn't have the expertise in this technique to make sense of. So that's what they're talking about there. Okay, so that's all for that. Let's see if we have anything in the chat. Okay. Do we have anything else we wanted to mention?
Well, I have a quick question. So, first of all, thanks for the presentation. But what kind of tools are we using to simulate some of these worms data? Like
which one for the paper, last paper?
you have shown in the paper. Like what kind of tool they usually use to use?
Let's see their methods look like here. They might mention it.
Okay.
Usually they use some sort of numeric simulation tool. I don't know if they actually mention what they're using here.
Okay. Actually, I was wondering, since it's growing, it might be growing. So are we interested in looking at different snapshots of the time? Like what would be the stages of a particular worm at a given time? Or we are interested in how it's growing, just like with the help of simulation? Because if we are interested in the specific features or like looking at the details about that, then like if we are looking at hypergraph representation, then I think the snapshot kind of model work better. So I'm not sure about that.
Yeah. Well, and Susan knows this, like with simulation, you have to sort of have a lot of computational power, because it's like if you simulate things down to like milliseconds or microseconds, it's really hard. Like when they do a lot of the protein folding work, where there are a number of groups doing the sort of simulating cells, and they're getting data, like molecular data, and they're simulating the molecular data, and molecular processes are like at like milliseconds and microseconds, and they're able to simulate those at very great detail. And so you can get that kind of detail, but then when you move up to the next scale, which is some cell process, you can't simulate, multiple, like, you can't simulate that process for like seconds because it's just too computationally expensive. So they have
Yeah,
to, but they do
exactly.
coarse graining, which is like they have to exclude a lot of detail to get a good approximation of that process. Sometimes you have to do
Yeah,
them in modeling. And so sometimes it makes sense to model everything like precisely. And sometimes you have to just kind of, coarse grain things and, just have a good enough representation for like a chunk of time or a chunk of space. And then that's, then you can have these larger scale simulations that have biological relevance. So.
Console might be able to model that, and ANSYS. So those are commercially available systems that are expensive. I don't know. You can often people make in-house ones, usually using Python. I don't know.
Or a MATLAB. I don't know if people have moved away from MATLAB.
Yeah, MATLAB. You can use MATLAB. Yeah.
yeah. Okay, thank you. I'll just check with some of those work. Yeah, I was curious to know because like some of the graph I would be also working like when we like look at different details, like the graph sometimes will have lakhs of edges or millions of nodes. So then it would be extremely intensive to even visualize this graph sometime.
Yeah, that's a whole different thing where you have like the graph layouts. You can actually make sense of the graphs. But then again, you might have like, you might not look at every node, right? So you just take a subset of the nodes that have like something to do with the structure.
Yeah, that's like more towards an open streaming graph or something like that, where we just interested in what aspect we are looking at. Yeah. Thank you.
You're welcome. Yeah.
I'm going to be trying MATLAB for the tensegrity structures.
Okay.
I have a program called Steady here. And hopefully trying Morpho and maybe, like I said, ANSYS. We'll see. I can get, for a student, you can get ANSYS for free, like beginner ANSYS.
Oh, yeah.
So, yeah. You can probably swing that.
Well, yeah.
Anyway, and I have Rhinoceros.
Oh, that's a tool or a model organism?
It's sort of a graphics interface modeling. They use it for architecture, so
all right.
Should be able to use it for engineering, although I'm not sure.
Yeah, sometimes they won't give you like the details.
Yeah, well, it needs to stand up. If you make it, it has to stand up. If it says it will in the model, that's engineering.
Yeah.
It must not fall down, no matter what. Yeah. Yeah.
Well, ideally, that's good for architecture too, I guess. But
yes, but you wonder.
Yeah.
So it just has to be good for engineering too, is what I'm saying.
Yeah.
Oh well.
Yeah. All right. Well, thanks for attending, everyone. Does anyone else have anything you wanted to add before we go?
So do you think that the angular deviation or the phase angle that we included, if we include that in while acquiring the structural features for the embryogenesis at each and every time point, this it divides into these two daughter cells, and this is the phase that we have seen. So including that phase information into that phase angle differences into the network modeling. Do you think that could give us a edge to understand the dynamics more better?
Well, we'd have, I don't know about the edge data, but it would give you like some spatiality to the graph. So it would be like,
Yeah.
you'd have like the edges would have like information about their divergence. So like if you wanted to say like something is maybe something belongs to two different modules, like you have like a mother cell, then there's a division event, and it moves out, and you have things that are maybe in separate clusters. You could say that you could say, okay, let's do a clustering algorithm on the points in space, their position, and then let's do the phase angle and see if they have a correlation. You can do all sorts of things like that, and I guess you could also use it as like edge information. You could say, well, this is a divergence between these two edges. Yeah. So if these two nodes were in the same hypernode and they shared hyperedges, those hyperedges are divergent in space, but they're part of the same hypernode.
Right.
Yeah, so that's another piece of information you could use.
Yes. Thank you.
Yeah, no problem. Okay, so let's call it a meeting. Thank you for attending. And if you're wondering about GSOC projects, proposals, those will be, the results for that we think we've released on like the 8th of May or something. So I'm not really sure what the process is going to be on that. So, but anyways, good luck to everyone on that. And we'll be in touch soon about, if you want to, if you don't get selected and you want to continue to work on what you were working on, we're open to that. But we'll be sharing details and all of that soon. So have a good week. Thank you.